This is a crystallization protocol designed to optimize the growth of a protein crystal while simultaneously derivatizing the protein with a phasing molecule to determine the structure of a protein via X-ray crystallography. To determine the X-ray crystal structure of a completely novel protein, crystallization conditions are optimized to produce quality crystals. In the case of a completely novel protein, heavy atoms need to be introduced into the crystal to produce a derivatized crystal. X-ray diffraction data is collected from the derivatized crystal and has to be computationally solved to produce an X-ray crystal structure. Crystallization and derivatization are both laborious processes. To address these bottlenecks, we have devised an optimized screening procedure that simultaneously achieves both of these objectives. This protocol uses a previously developed technique known as Random Microseed Matrix Screening. Crystals or crystalline precipitate is crushed up into microseeds, which can be used to start crystal growth in completely independent conditions. At the same time, we introduce a phasing molecule called I3C into the crystallization condition. This molecule provides a large anomalous signal which allows the structure to be solved via single wavelength anomalous dispersion phasing. For users unfamiliar with experimental phasing of crystal structures, we present a user-friendly software pipeline to partially automate the structure solution, tailoring it to use the anomalous signal from I3C. The first step is to create an I3C stop. Measure out 120 mg of I3C into a microcentrifuge.